Hello and welcome back. This will be the final part of the lecture on the combination of chapters 11 and 13. Uh, we had left off with the viruses and whether they were alive or not. The point I'm not sure I made clearly enough on the last section on viruses was this point about controlling viruses in the body. With bacteria, there are a number of different strategies, as we saw in chapter 10, because the structures are different. Things like the cell wall is different, the ribosomes are different. Um, yeah, so there's a number of targets having to do with protein synthesis. And so medications can target those differences with fungal medications. Will target the structure ergosterol because that's the one thing that's really that's different between fungal cells and our human cells. We use cholesterol, the fungi use ergosterol. So you get the idea. You're looking for targets of the physiology of those organisms that are different from the human physiology. And then the medications can be selectively toxic against the microbe. The problem with trying to fight off viruses is, one, viruses are tiny and they don't have a lot with them. So you'd think, they're weak. Yes, they're small, but because they're using all of our machinery, and the analogy I use, like use your toothbrush and you use the food in your fridge and use all your stuff, they're using our ribosomes, they're using our transfer RNA, they're using all of our machinery. So there's really nothing for us to kind of go after. So that's why there aren't as many antiviral drugs as there are antibacterial drugs, or what we refer to generally as antibiotics. They're coming, and we've certainly developed a bunch more with the COVID pandemic. But still, viruses are tricky. They're really tricky to get at. And so our best bet still with combating viruses is still vaccination and trying to expose the immune system to a version of the virus before we actually get sick with that virus. And we'll talk more about that in the coming weeks when we cover the immune system. Okay, now where we're going next is to a couple of things that are smaller than viruses. What? And they can cause disease in humans. They're smaller than viruses. We don't actually have a treatment for either one of these things. And so there you have it. So these are referred to as subviral particles. There's two main categories. One is viroids. Now notice this is different from virion. Virion is just the word that is used to describe a virus when it's not inside the cell. Viroids are smaller than viruses. They don't even have a protein coat around them. It's a tiny circular RNA molecule that needs another cell to make another copy of this circle of RNA. It's just weird. They're mostly known in plants. And in this class, we're all about public health, so we don't really care, except there's been one viroid that's been described in humans, and that's the hepatitis delta virus. And here's one of the things that's weird about it, and why we're not going to spend a lot of time on it in this class, is the hepatitis delta virus only affects people who already have hepatitis B virus. And so it's a parasite that only can work when this other parasite is present. So that's all we're going to talk about with viroids. I'm putting them on your radar because during your career as a nurse, other viroids may be described, and we might hear more about them in the treatment in the coming years. But right now, we got nothing. The next category, the second subviral particle, is even weirder. It's the weirdest of the weird. These are, it can be pronounced prion or prion. Both of them are right. This is what's so weird about this. There is no nucleic acid involved. So there's no DNA and there's no RNA involved. It's just protein. And what's even weirder about it, it's not even like we don't have that protein. All of us have this prion protein, but when it's in us and it's not causing problems, it's doing a particular job, it's fine. It's when it gets folded wrong, that's the problem. And there is no treatment for this yet, but I'm putting it out here because it is something that you may hear about, you may encounter a patient with this condition. It's devastating. This is lethal. And so we're going to wrap up this lecture on these next 4 slides. And I'm putting it out there just because you may encounter this condition in your career as a nurse. It's not unheard of. All right, now I'm changing the background color because this next topic, if it were up to me, this wouldn't have just been bundled in with the viruses in chapter 13. Like this just deserves not only its own chapter, I would put it in its own book. I mean, it's just so different from every other infectious agent. Yeah, prions are so different from other organisms and even from viruses. Viruses are huge and complex compared to prions. Pronunciation. All right, in the videos that I've included as part of the homework, it's pronounced prion. I say prion. They're both right. It's like the tomato problem. Tomato. That's what I hear in my head when somebody says prion. It's the British pronunciation. To me, tomato sounds right and prion sounds right, but both of them are acceptable. All right, so this is the other parasitic particle. These are the prions. And where does this name come from? First of all, the prion, the PR comes from protein or proteinaceous, and then the IN comes from infectious. This is what's so weird is we say it's not contagious the way, say, a virus can get from one host to another host through the air or by touch or something like this. These are infectious, meaning they cause a disease, but they have to be ingested. They have to be eaten. But there are exceptions. When we refer to the protein, because it's always one particular protein, and when I say one particular protein, I mean a particular sequence of amino acids that gets folded a particular way. That's what I mean by a protein. When it's working right, it is referred to as PRP, PR for prion and P for protein. So it's the prion protein, but it's the correctly functioning one. So we call it the cellular PRP. All mammals have it. Humans, all the primates, cows, sheep. Yeah, name a mammal, they have this protein. We don't, in fact, know exactly what it does, except it seems to be involved in neuron functioning, specifically the synapse between the neurons. Do you remember from your biology studies, proteins are made of amino acids, right? They're these long chains of amino acids. And that's the primary structure. Is what's the order of the amino acids? That's primary structure. And then secondary structure, we said there's some parts of it that are curlicue, like a telephone cord. That's the alpha helices. And then other parts that get zigzagged, the so-called beta pleated sheets. No big deal. That's part of the secondary structure. And then tertiary structure was how you fold those alpha helices and beta pleated sheets, fold them up. And finally, you might have quaternary level structure if you put more than one of these things together. Okay, fine. In the normal functioning cellular PRP, there are some sections of the strands of amino acids that have the alpha helices, the spirally part, and then they fold on top of that, and it has the correct 3-dimensional shape to do its job. In the defective version, that is what we call the prion. Where's my cursor? There it is. The prion PRP. in those sections where there's supposed to be alpha helices, it's the same amino acid sequence, but instead of being oriented like this, it gets oriented like a zigzag, like a beta pleated sheet. If you need a way of picturing this, if you know what a curling iron is, a curling iron would do a curl, and somebody's coming to be in the view. This is my child. And so there's a curling thing that's an alpha helix. And then there's a crimping iron that would make the hair look zigzaggy. That's a beta pleated sheet, zigzaggy instead of curly. Okay. So for reasons that we don't fully understand, sections that are supposed to be alpha helices, supposed to be curlicues, they turn into this zigzag beta pleated sheet. And once they're in that beta pleated sheet conformation, one, they don't do their job anymore. So the cell keeps trying to make more of that protein, but it keeps folding wrong. And so the second problem is it's resistant to proteases. It's resistant to the enzymes that break things up that aren't working. So you can recycle those bits. So instead, you get a buildup of these disease prion PRPs, and then the cells end up dying. It's tragic. It's really bad. And remember, I said it's in the neurons. So it's in the parts of the brain. And so we have a bunch of these collections of these prion PRPs, the cells actually die off. And I'll show you what that looks like. Oh, here's the curlicues and the zigzag. All right. So it's a protein that gets placed into the cell membrane. It has multiple parts to it. And some of these sections are supposed to be the curlicue, alpha helices. And instead, those same sections, they still have the same primary structure. They still have the same amino acids in sequence, but instead of arranging themselves like this, they arrange themselves with these long zigzags, the beta pleated sheets. And you might think, big deal, like who cares? But the problem is now that this is misshapen, it can't do the job that it's supposed to do. We still don't know exactly what that job is. We just know that it's not able to do that job because the cell keeps calling for more of it. You still get, the signal to transcribe this gene in the nucleus and then translate it on the ribosomes and make this protein. And the problem is it keeps getting misfolded. And so we still don't have functional protein. So the signal goes out again and you keep making more and more and more of this stuff and it keeps getting folded wrong. And that's what ends up killing the cells. All right. Before this was finally described, before we knew what was causing this problem, that it was this one protein, this problem was described in humans in 4 different ways. And in a bunch of different animals, it's been described. It turns out it's all the same thing. It's all the misfolding of this one protein. So in human diseases, Creutzfeldt-Jakob disease is what it's referred to as. Let me see if I can show you. Yeah, so this is supposed to be an image of a brain of a cow. And you've probably heard of mad cow disease. This is what we're talking about. Mad cow disease is due to the misfolding of this cellular PRP protein. And it causes the death of neurons in the brain. And when these neurons die off, they leave these open areas in the brain. That is visible on imaging, but it's not visible until it's quite advanced. So it can take 4, 5, 10, 20 years for this thing to get to the point where it's actually identifiable as this problem. So the incubation, as we say, the latent period of this infection where the person maybe doesn't know that there's a problem yet is years to decades. And what starts looking like is these, where is my cursor? These bubbles, if you will, in the brain. It looks like a sponge. And so it's spongiform, meaning formed like a sponge. Encephalo has to do with the brain, and pathy is disease. And so spongiform encephalopathy means looks like a sponge here in the brain, and then depending on what animal you're talking about, here bovine, meaning having to do with a cow. In lay terms, this is called mad cow disease. We know that this can spread to humans if humans eat this misfolded protein. So people don't catch it from touching this thing or from breathing this thing. They catch it from eating an infected cow, specifically eating brain matter, but it can be in other parts of the animal. Okay, and why it started spreading in like slaughterhouses or ranches that had cows living there is because sometimes you're trying to grow an animal and you're trying to grow the animal for the protein of the animal. And so sometimes these vegetarian animals are given, or these herbivorous animals are actually given some animal meal. And yes, it's cannibalism, but basically the bone meal can be crushed up and added to their food. It helps them to grow, unless there's some of this PRP protein, this prion protein already in there, and then the cows can have this problem, and then it can spread to human populations who eat those cows. Okay, that's the version that we were first introduced to in large numbers in the 1980s and 90s. It's called variant Creutzfeldt-Jakob disease, or VCJD disease. All right, that's enough about that. What else did I want to tell you about this? A few things. There's a great video as part of the homework that kind of walks through all of these different categories and which part of the brain gets affected. Like Creutzfeldt-Jakob disease affects the cerebrum, but one of these other diseases affects the cerebellum. Fatal familial insomnia is a condition. This one's a little bit trickier. It turns out that people who have fatal familial insomnia actually have the genetic code for the cellular PRP, that there's a typo in that gene that causes this protein to be more likely to change shape and to become the prion protein. And once that starts happening in somebody's middle age, it advances to this terminal situation where the person cannot sleep and they end up falling into a coma and dying. It's awful. And finally, the example Kuru, this one was in New Guinea, and there was some ritual cannibalism. It was part of a cultural grieving process where the brain matter of the relative was prepared for ceremonial consumption after their death. And then the people who had consumed that brain matter years later would have this laughing problem, where they uncontrollable laughter, and to the point that they would eventually die. I mean, the laughter was kind of a symptom of what was happening here in the brain. We see the same problem happening in sheep. It's known as scrapie because the behavior that the sheep exhibit when they have this condition is they rub themselves raw. They scrape their skin against any surface. So it's called scrapie. It's also been seen in mink, small rodent that's used for its pelts. Chronic wasting disease. This is also of concern in deer and elk populations, where this has been identified. Yeah, it is bad news, and we don't know what to do about it yet. So the problem always comes down to spongiform encephalopathies. That's a fancy way of saying the brains are getting holes in them. It looks like this. These large vacuoles form because the cells that would ordinarily have been there are dying off. So they give this characteristic spongy appearance. There's bovine spongiform encephalopathy, so-called mad cow disease. Then the humans that end up getting the disease from having consumed the infected cows. This is variant Creutzfeldt-Jakob disease, or VCJD. And then, as I mentioned, this one, Kuru, was isolated to human cannibalism in New Guinea. And again, the cannibalism wasn't like people going out and killing other people to eat them, but the ceremonial preparation of the brain matter of the revered relative. And that was ingested in the 1950s. This was outlawed completely, and Kuru has gone away. All right, so it's transmitted by ingestion of the infected tissue, could be from transplantation or contact with the mucous membranes with infected tissue. And then a small fraction of the cases of this prion disease can be traced back to this gene mutation. That's the fatal familial insomnia. It affects the thalamus part of the brain. Here's the thing. Remember how we talked about, getting rid of bacteria is tough, and you have to wash things and clean them and autoclave them and do whatever. None of that fixes prion. Like, you can't get rid of the prions. And so the only fixes that we've found are incineration, just burn the thing, or autoclaving, and not even just autoclaving. Autoclaving is like basically high pressure, like cooking something in a pressure cooker, but like industrial strength pressure cooker. But it can't even be in water. It has to be sodium hydroxide. That's a very, very high pH, really damaging. So we don't even have a guess at how to do a treatment for this. So at present, there's no treatment. Why I'm bringing this up, though, is that you should stay tuned for information about this throughout your career as a nurse in the coming 20 to 30 years. There are going to be advances in this. And the person who first described this won the Nobel Prize, because this was the first case that was ever described where there was transmission of a disease and there's zero genetic information. There is zero DNA, there is zero RNA. It's just proteins. And it's not even proteins making other proteins. It's the presence of a misfolded protein somehow seems to cause the refolding of what was a functional protein and makes it into this poorly folded protein. And that's what causes the disease. And I say stay tuned because if I were a betting man, I would say there is something that we have not identified yet. There's a missing piece to this puzzle, because there's no way that this protein being folded this way is causing that protein to fold that way. It's like the whole one bad apple, spoils the whole bunch. There's something missing in this story. And if you're looking for a way to get yourself a Nobel Prize, you figure it out, because there has to be more to the story than this. All right, and so that is the end of the subviral particle part of this lecture, and I hope you have a great week.